Okay, so good evening. Welcome to week four live interaction session. So this week's topics mainly centered around the process of mitosis and the following procedure, which is of cytokinesis that completes the process of cell division, and a bit of introduction into the role of centrioles and centrosomes and how they have evolved over a period of time. So before we get into discussion regarding these topics, we'll first go over the questions that were part of the previous week's assignment. And if there are any doubts regarding the same, you can bring them up and we'll discuss them as well. So for week three, which was mainly centered around the cell cycle, the S phase, the process of replication, prokaryotes as well as in eukaryotes, we'll go through the questions one by one. So the first question looked at which of the following statements are incorrect about the S phase of the cell cycle. The options talked about A being DNA duplication occurs in this phase, which is certainly true. The second option is that once the cell crosses the restriction point in G1 phase, it irreversibly enters the S phase, which is also true. It was considered to be the commitment step or the step that marks that a cell has now decided that it will go into division and therefore it is going to replicate before it undergoes the process of division. Third being that certain external stimuli can reverse the progression of S phase even after the cells have crossed the restriction point. So this option is of course not something that is going to that will be allowed by the cell. That is because once the cell has committed to the S phase, and they have crossed the restriction point, which is what we discussed in the previous option, they cannot revert back. So no external stimulus can actually reverse the same. And the only time where the external stimuli can allow it is when a cell is trying to decide when it is transitioning from the G1 to the S phase. Only during that period, external stimuli, particularly in the form of mitogens, can influence whether a cell will undergo division or not. And if it is in a situation where the cell is starving and it does not have enough resources, such a cell will not be allowed to go into division. So that is the point where regulation can actually take place and not beyond that. So the third option is certainly correct. And in the fourth, we are being told that in East, progression through S phase requires the CLB5 and CLB6 cyclins, which is true. So the incorrect statement was option three. So for the second question, the ORC or the origin recognition complex binds the replication origin during which phase of the cell cycle? So the ORC binds the replication origin. First option is at the late G1 phase, throughout the cell cycle, early S phase, or throughout replication. When does it bind? So we have understood and we have seen that the recognition complex is always bound. It always occupies the it always occupies its binding site throughout the cell cycle in order to ensure that it can only be that replication can only be initiated once throughout the cell cycle, and it does not during one division it does not undergo multiple replications because that will lead to a number of anomalies. It will lead to multinucleated cells, which is something you do not want, and can also lead to neoplastic transformation as well. So therefore, the correct option is option B, that it binds the replication origin, which is its binding site throughout the cell cycle. Option three, which of the following sorry, is incorrect about DNA replication? The first option is a primer template junction is necessary for DNA replication to begin. So this is true. If you remember, we had seen that DNA replication, which is being carried out by DNA polymerase, it is an enzyme that cannot initiate its activity in a independent manner, which means it cannot start ab initio. That is the terminology which is normally used. So it cannot start from anywhere. It needs that available 3' OH end, and it needs the template to also be present. So that primer template junction is where it would be able to start and then carry out its activity, which is why you need Pol alpha primase to come in and first present the RNA primer or prepare the RNA primer, onto which DNA polymerase can bind and then execute its activity. So Option A is correct. Second option is that the extension of the primer occurs at its 5' end. That is certainly not true, which is just what I said, that the enzyme needs a 3' OH end to be accessible for it to begin its activity. Therefore, option 2 is incorrect. If we still look at the other two options, it says that DNA polymerase is required to catalyze the formation of the phosphodiester bond. It is certainly true because it keeps on adding the enzyme. As the name polymerase indicates, it is responsible for recruiting or adding more and more DNTPs onto an elongating chain. And it does so by removing two phosphate groups and creating phosphodiester bond between adjacent entities, and therefore extending the entire deoxynucleotide chain. So its job is to create new phosphodiester bond and extend the DNA backbone. Then, fourth option was that a correct base insertion is ensured by complementary base pairing with the template. So while to a large extent that is required, it is not the only way that you can ensure that there is correct base insertion. There is an entire machinery which is present for this purpose, and not even just one machinery, it's multiple molecular machineries which are present inside the cell. Commonly, if you are interested in it, you can look it up. They're commonly known as MMR and BER, which is mismatched repair machinery, and the base accession repair machinery. So they have been recruited within the cell to make sure that whatever nucleotides are present on the template strand, with respect to that, the newly synthesized strand should always have the correct base being inserted against it, that there should always be complementarity, and any um, misinsertion during the process of replication can actually is actually what you understand as mutation, and such mutations can have very far-reaching consequences. And it is of course another way in which a cell keeps its genomic integrity intact and ensures that the process of replication is carried out smoothly. Now, while polymerases themselves also have some exonuclease activity where they can remove the last inserted base in case there is no correct insertion, or there is an incorrect insertion that has happened, it is not foolproof, and still polymerase slippage and polymerase can make its mistakes. Therefore, all these additional machineries are required. So option 4 is not entirely correct. Therefore, from these, option 2 is what is correct with respect to the question, that extension of a primer does not occur at its 5' end. For question 4, we were asked that a phosphodiester bond is formed between which of the following? So which are the two entities which are forming the phosphodiester bond that is being carried out or is being catalyzed by the DNA polymerase? So if you remember, um, the template or the initiation point for polymerases is 3' OH, and what it will bind to, or what it will catalyze with, is the phosphate of the incoming nucleotide. So 3' OH of the preceding base will be linked or will form a bond with the alpha phosphate of the 5' carbon of the incoming deoxynucleotide. So based on this, because all of the options are quite similar, I'm not reading out all of them. The correct option will of course have 3' OH of the preceding base with the alpha phosphate at 5' carbon of the following base. So option A is correct among them. If there is any confusion, you can look at the diagram that we had seen in the previous class, where the chain of nucleotides that is being formed, the order in which it is being formed, you can look at that. And of course, you can look at reference textbooks as well for more clarity. Then for question 5, Okazaki fragments are which of the following? So if you remember how we had discussed that the process of replication is extremely different for each of the two strands, for the leading and the lagging strand. Not just the discontinuity or the continuous or the semi-discontinuous nature of replication, but also the way in which polymerases are recruited, the way in which primers are recruited, the rate of synthesis, and a number of other factors. So the way in which that the cell ensures that the rate of replication for the two strands does not vary by a lot is that it ensures the formation of short segments of deoxynucleotides, which are then ligated with each other, and it forms a complete DNA strand, complete daughter DNA strand for the lagging strand template. So Okazaki fragments, therefore, are short segments of DNA strands that are made of 100 to 2000 nucleotides, which is of course option A. If it was an RNA strand, then that would have to be removed, because like we have understood that the RNA primers are always removed from the cell, and therefore it would not serve the purpose. And therefore option 2 is not correct. And of course option 3 and 4 are also not 3 and 4 are also not true, because they are not polymerase mistakes. They are an evolutionary mechanism that the cell has come up with in order to make sure that the process of replication is maintained at a similar level, or at a similar, the dynamics of the process of replication are being maintained for each of the two strands during the process of replication. So option A in this regard is correct. Um, then for question 6, we are asked to pick the incorrect statement about prokaryotic DNA replication. Now, most of the discussion that we had done, I think, was centered mainly around prokaryotic replication only. And we introduced the differences with eukaryotic replication in the last interaction session. And then we saw a comparison between bacteria, archaea, and eukarya. So for prokaryotic DNA replication, which of the statements is not correct? First, that prokaryotic chromosome is usually circular. That is certainly true. We have seen that most prokaryotes, they possess a circular genome, which is why they do not have the problem of telomeres or shortening of chromosome ends, etc. So their genome is also often, for example, if we talk about bacteria, bacterial genomes are majorly in the form of plasmids, which are otherwise also known as cccDNA, or covalently closed circular DNA. And therefore, the way in which replication is performed in their case is quite different to that in eukaryotes. Second, prokaryotic chromosomes have multiple origins. That this is, of course, not true. And the reason that it is not true is because they have a relatively much smaller genome compared to the extent of information that lies in a eukaryotic genome. Therefore, only one replication origin from one particular point, leading to two replication forks completing the entire replication process, eventually leading to fall off, is sufficient to replicate the genetic information that is there in prokaryotes. In contrast, for eukaryotes, A, the fact that we don't have a circular genome, and B, because we have extensive information, or a much larger genome compared to that of prokaryotes. Therefore, to make sure that the process of cell division is not limited by the rate at which DNA can be duplicated, we have multiple origins in place. So option 2 is incorrect for prokaryotic replication. The other statements are that replication initiation requires initiator protein and primosome. That is true. The INR and the primosome is required because of the way in which polymerase functions, like we just discussed, and replication proceeds bidirectionally. That is also true. If you remember, we had looked at the theta model, that from one place the origin starts, and then eventually the circle starts looking like a like the Greek symbol theta. So option 2 is the answer for this, because it is the incorrect statement for prokaryotic DNA replication. If there are any doubts, please interrupt me at any point of time, and I'll try to clarify it for you. Uh, in question 7, we were asked that Okazaki fragments are formed in which strand during DNA synthesis. So we just had an elaborate discussion of the way in which Okazaki fragments are formed and the reason that they are formed. So of course, the need for them is to be formed on the lagging strand, and the reason for it is because of the way in which polymerases can move and the way in which they can function, and the fact that there are multiple polymerases that are recruited in order to complete the replication of the lagging strand. So the correct option in this case is option number 2. For question 8, the unwinding of the parental DNA helix during replication is carried out by the replicative DNA helicase, called which of these following? So the easier way to remember it is the order in which these individual proteins are recruited during the process of replication. And in fact, that is, I think, the way in which the nomenclature of these proteins was also done. Since helicase comes second, therefore, the the other way in which DNA helicase is also referred to is DNA B. So this is a very direct question. It's a memory based question. So DNA B helicase is recruited during replication for unwinding of the parental helix. For question 9, we were asked which of these following statements are incorrect about eukaryotic DNA replication. First being that eukaryotic chromosomes are linear. They have multiple origins of replication. All the origins fire simultaneously. Sorry, excuse me. Or DNA replication happens only once per cell cycle. So for eukaryotic DNA replication, if you let's go by the statements one by one. First, the eukaryotic chromosomes are linear is certainly correct, which is what leads to the end replication problem, like we just discussed, and that is why the concept of telomeres has come about in order to make sure that during each subsequent replication, the genetic information from the ends of the chromosome are not lost. Second, that they have multiple origins of replication. Certainly, eukaryotes have multiple ories, like we just discussed, in contrast to prokaryotes that only have a single origin of replication. For option 3, all the origins fire simultaneously. So this is not necessarily true. It does not happen in a case of eukaryotes that all of the origins should simultaneously fire. It is not required. It depends on whether the recruiter complex has assembled and has started replication at each of these origins or not. And option 4 is that DNA replication happens only once per cell cycle. That is also true, like we just discussed. And there is an elaborate mechanism that is kept in place to make sure that during one cell cycle, during one S phase, DNA replication is only initiated once, so that a cell that was previously had a ploidy of n can only duplicate to form 2n, during which would be followed by the process of division, and that would lead to two daughter cells each having n ploidy. So it is essential for the cell to maintain that only one round of replication is initiated in one cell cycle. Finally, in question 10, we were asked that in yeast, the leading strand is synthesized by which of the following? DNA Pol beta, Pol delta, Pol alpha, or Pol epsilon. So for yeast, the leading strand is synthesized by Pol epsilon, which is what was seen during the process of eukaryotic DNA replication in our previous live interaction session as well. So Pol delta and Pol epsilon majorly carry out the process of replication. In case of eukaryotes, particularly elaborate studies have been carried out in yeast, and among them, polyepsilon is what controls replication for the leading strand, which is what the question has talked about. So with this, the questions that were a part of the previous week's assignment come to a completion. So if there are any doubts, you can bring them up, and I'll try to address it. And then we'll look at the week 4 topics that were included in these lectures. So just a second. Okay, so if there are no questions, you can bring them up any point later during the class also. So for now, we'll proceed with a discussion of the video content or the lecture content for this week. So first of all, the overall idea that was looked at in this week was to go over the process of mitosis and the regulatory steps that operate in the process of mitosis, and what is the significance of it, how is it controlled, and what is it eventually followed by, which is cytokinesis. So basically that is what has been looked at. Now, before I get to the details of what was part of this week, what is something that you need to clearly understand is that there is a difference between the processes of meiosis and mitosis. While they might sound similar and might seem to like eventually end up doing the same thing, what needs to be understood is that the ploidy of the cells that are eventually being produced as a result of these processes is different. So meiosis and mitosis are both forms of cell division, but the way in which the chromosomes are segregated, they are duplicated and segregated, leading to the generation of daughter cells is different in either case. So in other words, how it is commonly described is that the process of meiosis is reductional division, whereas the process of mitosis is equational division. Why is it named so? So reductional division means that there is a reduction in the number of chromosomes that were present in the parent cell compared to what is present in the daughter cell. However, for equational division, the number of chromosomes that were in the parental cell are equal to the number of chromosomes that are in the daughter cell. So there is no reduction in case of mitosis, whereas for meiosis, there is difference in the same. So the process of meiosis normally takes place for cells for germ cells, otherwise what you understand as gametes. So during the process of development of or embryogenesis, there is this division in a way such that the gametes that are produced in all individuals, for example in our case, the sperm and the ovum, they are not diploid, whereas all other cells in our body are diploid, which means that they have two copies of chromosomes, or the entire genetic information of a cell is present in two copies in all cells of our body, except for cells that are haploid in nature. So these haploid cells are mainly the gametes. There are some exceptions to this as well. For example, RBCs, they do not contain a nucleus. They are post cells, so they do not contain such genetic information. Sorry. Um, so yes, so these are haploid cells. In contrast, all of the other cells in our body are diploid in nature. So for production of these gametes, the process that they have to go through is meiosis. Whereas for production or for duplication or division of all other cells in the body, what the basic process that they have to go through is mitosis, which means that from cells that are that carry N ploidy will divide to form cells that have N ploidy, versus for meiosis, cells that have 2N will divide to form cells that have N ploidy. So this is the major difference between these two processes. However, what needs to be kept in mind is that the basic sub-steps or the processes that or the arrangements that the cell goes through during this process of division are largely similar, which means that there are the common steps that exist between the two are prophase, metaphase, anaphase, telophase, and then finally cytokinesis, which is division of one cell or one parent cell or one mother cell into two. So this basic steps, these steps are being conserved in both of these processes or both of these modes of replication or cell division, not replication, cell division, that is both in case of meiosis as well as mitosis. So with this in mind, let us look at the process of mitosis. So what you need to understand is that there is, like there was a point at which the cell decided that it is now going to enter the process of replication, which was once again regulated by the crucial complexing of cyclins as well as CDK that basically regulate the entire cell cycle by their expression levels. The process of mitosis is also initiated and controlled by the cyclin CDK complex. Just a minute. So like you can see here, the process of mitosis will be regulated in particular by the mitotic CDK or mitotic cyclin dependent kinase, that is otherwise known as MCDK. So MCDKs, I think the presentation is getting loaded, maybe just a second. Yeah. So like you can see in the diagram, CDK1, it associates with the M cyclin, that is the mitotic cyclin, as the levels of mitotic cyclin gradually rise when the cell enters the M phase. The resulting complex gets phosphorylated by a CDK activating kinase, which you can see here as CAK, represented in blue, and on a pair of inhibitory sites by the V1 kinase. If you remember, during our initial cell cycle interaction session, we had understood that there are multiple or the role that phosphorylation plays for cell cycle can be both inhibitory as well as activating in nature. So this can depend upon the context in which it is supposed to function, and the effect that it will have can also differ from the position that the cell currently is in the cell cycle. So if you look here, the V1 kinase in particular is working in an inhibitory manner, or it is initiating two inhibitory phosphates or one inhibitory phosphate, whereas the activating phosphate is being brought about by CDK activating kinase. Now, because of the presence of these inhibitory phosphates, the MCDK enters into a inhibitory or inactive state, which gets oh, sorry, which gets activated at the end of the G2 phase. If you remember the order in which the cell cycle used to perform, it is first G1 followed by the S phase, which is followed by G2, that is the preparatory phase for mitosis, followed by the M phase. So the pre-mitotic phase is the G2 phase. Now during G2, the inactive MCDK complex is getting activated, and the activity is being regulated by CTC25 phosphatase that removes the same. So for its reference, you can see the diagram at the bottom that there is a cyclin and CDK complex. Initially the activating phosphate, or initially we can see that there is one phosphate that is present, and that is resulting in an active cyclin CDK complex. However, the action of V1 kinase introduces an inhibitory phosphate, due to which that cyclin CDK complex is no longer functional. However, CDC25 phosphatase comes in, removes this inhibitory phosphate, and activates um, the MCDK cyclin complex. So you can see that is exactly what is happening during this phase as well. In addition, the MCDK, CDC25 also further gets stimulated by the active MCDK, and that is leading to a positive feedback loop, which you can see on the top. And the feedback is also enhanced by the ability of MCDK to inhibit V1 kinase. So what this effectively ends up doing is that once the stimulus has been received, that now the cell is going to enter the process of cell division, or it is going to enter the M phase, then the cell brings into favor multiple mechanisms that are going to ensure, or it initiates a cascade that is going to ensure that the cell enters the M phase and stays in that M phase. So there are positive feedback loops that are enhancing the same. So one way is to inhibit V1 kinase, the other is to stimulate or activate the CDC25 phosphatase. So that is how entry into the M phase is being regulated. So following this, sorry. Following this, what happens is that the cell has now decided that it is going to enter the M phase, and within the M phase, we'll look at each of these steps one by one. All of the diagrams, or most of the diagrams that you see here, have been taken majorly from three books. Most of the diagrams or most of the schematics are from Bruce Albert's Molecular Biology of the Cell. I have also referred to it previously for the cell cycle lectures as well. And apart from that, you'll find just one diagram, but it is a wonderful book for understanding genetics, which is Russell I Genetics. I've also added it as a reference. And some information is also from Gerald Karp's cell biology book for your understanding or reference. Each of the references have been cited below also for your better understanding. Okay, so coming back to each of these steps of the process of mitosis. So like we said, the first step is certainly prophase. So like you can see, on the left, you see a schematic representation. On the right, you see an actual immunofluorescence image. Immunofluorescence in this case means that it has been stained by antibodies for specific proteins. What you see in green is the microtubule network. We'll come into details about it. Microtubules are part of the cytoskeletal network, which is present throughout the cell. And initially, you can see that it is rather disorganized in nature. And what you see at the center, stained in red, are chromosomes. And this is representative of the entire cell in the initial stages of cell division. So coming to the figure legend, you can see that at prophase, the replicated chromosome, which has already replicated during the S phase, these replicated chromosomes each consist of two closely associated sister chromatids, which you can see on the left schematic image that they are being represented in brown, and they are being attached at a certain point by some red colored protein, and that is understood to be the centrosome or the kinetochore of the chromosome that is keeping these two sister chromatids associated with each other. So in the initial stages, they start condensing. What is the meaning of condensing? It means that prior to the process of cell division, or prior to prophase, the entire nuclear information, or rather the genetic material of the cell, is present in a disorganized manner as chromatin. Whereas when it is decided that the cell is going to duplicate, it starts condensing or it starts gathering all the information into an organized manner, or forms definitive chromosomes that would be easier to understand, duplicate, manage, and equally divide among the daughter cells. So that occurs during the process of cell division. Now, this condensation is majorly, it's brought about by a large number of proteins, but it is crucial because it is going to allow equal division or equal segregation of genetic information among two daughter cells. So, coming back to the legend, it says that outside the nucleus, the mitotic spindle assembles between the two centrosomes, which have replicated and moved apart. Um, it's very vaguely you would be able to see that in the immunofluorescence image as well, that there are two distinct points that are very dense or that have intense green signal, and that is representative of the two centromeres or centrosomes from where the mitotic spindle has started to form. It is not very extensively organized, but it is present. Um, Also on the schematic, you can see that only three chromosomes have been shown. That is only for simplicity purposes, but for each organism, depending upon the number of chromosomes, it will vary. Additionally, what you can also see is that the nuclear envelope in the initial phase, that is during the prophase, it is still present. You can still see the nuclear lamina, which is in place. It has not been stained, which stained separately, which is why you're not able to see it in the immunofluorescence image. But the nuclear envelope during this stage is intact, and this nuclear envelope is otherwise also known as lamins. And if you remember, we had discussed that also, that there is dissociation of the lamina, nuclear lamina, in order to allow the cells to proceed through the M phase. Then comes the next phase, which is pro-metaphase. Now, pro-metaphase, as the name indicates, is representative of a pre-metaphase stage, which is the stage just before metaphase. So pro-metaphase starts with the abrupt breakdown of the nuclear envelope. So like you see in prophase, there is an intact nuclear envelope which is present, but chromosome condensation has taken place. This is followed by breakdown of the nuclear envelope or disassembly of the nuclear envelope, which is what we just discussed. This allows for each of the chromosomes that are present and that have now condensed and organized themselves appropriately to attach to the spindle microtubules. So that is necessary because the nuclear envelope under normal circumstances would prevent anything, would prevent rapid entry and exit of any other proteins. For the mitotic spindle to be able to attach to the chromosomes, the nuclear envelope has to be dissolved and chromosomes have to be attached and they have to be segregated. So what you see in the schematic also is that there are fragments of some little fragments of the nuclear envelope. You are seeing that the chromosomes are taking a different shape. That is because the central portion, which contains the kinetochore, is where the spindle is attaching. And you can see that they are localized in different positions and they are ready for segregation to take place. A similar representation is also seen in the fluorescence micrograph. Then comes the metaphase, which is the next stage, followed by which follows pro-metaphase. So metaphase, as I think we have previously also discussed, is most distinctly understood by what is known to be the metaphase plate. So at the center of a dividing cell during the metaphase, all of its chromosomes are aligned and along the equatorial plane. What is meant by the equatorial plane is that it is the center. If you take the Earth into consideration, its equator is the central um, line that runs throughout the spherical structure. So similar to that, for a cell, if you consider it to be a sphere, then what would run around the center of it is its equator, and this equatorial plane is otherwise also known as the metaphase plane or metaphase plate. So along this plate, all of these different chromosomes are being aligned um, in a particular manner, which also has a definite term, which we'll come to eventually. So this is the distinctive recognition point of a metaphase plate. So coming to the legend, at the point of metaphase, the chromosomes are aligned at the equator of the spindle, midway between the two spindle poles. So you can see that the in the fluorescence micrograph, the red representation, which is for chromosomes, shows that they are all aligned at the center, and you can see huge number of microtubules which are getting associated with these kinetochores. And what you see in yellow is not a separate stain for it, it's just the overlap between the two, because these are positions where both the DNA as well as the microtubules are fluorescing, and that is leading to generation of a different color. So the kinetochore microtubules are attaching sister chromatids to the opposite ends of the or opposite poles of the spindle. Now this is again very significant. Why is it significant? If you looked at the lectures very carefully, you would have seen that the orientation or the arrangement of chromosomes during this process of metaphase also has a very crucial role. Uh, the way in which kinetochore microtubules are attaching to the chromosome has a significant role to play in it as well. This is because the sister chromatids, which if we represent, say suppose in this manner, if I show it as two sister chromatids being attached at the center point, which is the kinetochore, then these two are present in a manner which is known as back to back. It is not that they are present like this or like this. They are present in a way such that the kinetochore or the site to which the microtubule is going to attach is facing the outside. Why is this so crucial? That is because after alignment has occurred, it should not be such that the cell is unable to distinguish between the two sister chromatids, or the site of attachment of the microtubules is not clear, as a result of which both the sister chromatids, say suppose they are in this manner and attachment does only occur at one end, then the migration of both sister chromatids would occur only towards one mitotic spindle. Whereas if it was oriented in this manner, then its migration would only be in one direction. That would be extremely detrimental to the cell, and that leads to disorders, chromosomal disorders, which are otherwise also known as non-disjunction diseases. I think we discussed that as well. One common example of it is the Turner syndrome, which leads to XO. It's a disorder of the X chromosome. So one daughter cell would not have any chromosomes, and that is a null cell. So such correct orientation and correct arrangement is very crucial and is regulated by a large number of mechanisms, which is something also that you came across in the lectures. So following that, the next phase, which is the fourth phase, and again one of the most crucial phases of the mitotic cell cycle, is the anaphase. So after all of these cells had aligned during metaphase, the next step is to carry out the segregation of the two sister chromatids at two different mitotic spindles for subsequent separation into two cells. So the sister chromatids, they synchronously separate to form two daughter chromosomes, and each is pulled slowly towards the spindle pole that it is facing. The kinetochore microtubules keep on getting shorter, and the spindle poles also move apart. Both processes contribute to chromosome segregation. So this actually is described in a somewhat sub-split manner. There is anaphase A and anaphase B. So during anaphase A, the chromosomes start moving, the sister chromatids, they start moving in opposite directions, and that is also accompanied by shortening of the microtubule kinetochore microtubules that were attached to the chromosome. So the movement is occurring because the microtubule that was attached has started to dissociate. However, in anaphase B, the two mitotic spindles, which are the points of contact inside the cell. So you look at it as two anchors. There are two main anchors inside the cell from which these huge networks are originating. So these two anchoring points, they start to move apart, and that is creating further tension and further segregation among the genetic material, which is going to divide into two cells. So there is anaphase A and anaphase B, which are regulated in these sub-steps. So even in the schematic representation, you can see a similar arrangement. And often this is one of metaphase and anaphase are one of the easiest phases to look at if you look at cells under the microscope. So one common experiment that is often done is that onion root tip cells are taken, and they are like the squash preparation is made. So you take onion roots and you prepare a smear of it or a squash of it by some specific chemicals that are required for it, and then you stain the chromosomes that are inside these onion root tip cells. And the reason that root tips are taken is because these are actively dividing cells, because you would then be able to see cells in different phases of the mitotic division or the process of mitosis. So there, the easiest phases to distinguish are A, for those cells that are in metaphase, because you'll be able to see cells that are aligned at the center, and B, the cells that are in anaphase, that is because they would have chromosomes that either have a V shape or a U shape because they are being pulled apart by the chromosomes that are by the mitotic spindles, which are connected to them by kinetochore microtubules. They are being pulled apart towards the opposite ends. So that is what we see in anaphase. Following this comes telophase. So during telophase, the two sets of daughter chromosomes, they arrive at the poles of the spindles and now they start to decondense. So if you look at the schematic, you would be able to see that there is some reassembly of nuclear envelope which has started to occur. Again, this is not visible in the micrograph because they have not specifically stained for it, but it does take place. So now once the segregation of chromosomes at two opposite poles has occurred, there should again be reassembly or processes that would try to reinstate what was previously in place. And the first step that occurs in this direction is that the nuclear envelope starts to reassemble, which completes the formation of the two nuclei, and it effectively marks the end of mitosis, that now cell division is coming towards an end. So once the nuclear division has occurred, which is karyokinesis, the next step is cytokinesis, and that is division of the cytoplasmic material, and this division will begin with the initiation of the contractile ring. So we'll come to that a little later. So what you see in the schematic is that there are a set of daughter chromosomes that are present at each spindle pole. You can see the red colored kinetochore, which is facing the mitotic spindle, or the centrioles that are present at the mitotic spindles, and you can still see some microtubules which are relatively longer. That is because they are interpolar. They are not kinetochore microtubules, which means they did not attach to the dividing chromosomes, but instead they are present between the two poles and they maintain this segregation between the two. And finally, in this beautiful image on the right, which is for cytokinesis, which beautifully stains for the microtubules, you can see that cytoplasm is being divided into from one into two by a contractile ring for actin and myosin filaments, which pinches the cell. So you had one large cell, it starts invaginating and then it pinches off into two cells to create daughters, which each have one copy of each of the chromosomes with an intact nucleus. So that is what you see in the schematic as well. But what you see as a difference is that they will again start to decondense. The intact chromosome or the condensed compact chromosome that was initially in place will again be resolved, and it will again form the loose chromatin, the uncondensed chromatin, which will allow the cell to remain in this interphase before it receives another cue for cell division. So there is reformation of the interphase area of microtubules that are nucleated by the centrosome, and there is a contractile ring which you can see at the center, which creates the cleavage furrow from where the cell will divide into two. And this is what the process of cytokinesis effectively does. So cyto is for the cytoplasm or cell, kinesis is for division, versus karyokinesis is for karyon, which is nucleus, and kinesis is again for division. So nuclear division and cytoplasmic division are being regulated at these two different steps. So with this, we understand what is happening during the process of cell division. Next, what you see on the left is a representation of the different ways in which a chromosome, the ways in which a chromosome can appear under the microscope. So so what you see is that the along the length of a chromosome, when it is in its condensed state, the primary constriction, what does that word mean? Primary constriction means the first, if you look at a continuous cylinder, say for example, this is a continuous cylinder, right? Along its length, when there is a first invagination or a first disruption from the continuity, or there is a constriction that is present, then that would be understood as a primary constriction. Why is it known to be so? That is because when you look under the microscope, which is how chromosomes were initially studied, what it appeared as was that along the length of the entire cylindrical like structure of chromosomes, um, there was a slight bit that appeared a little pinched or a little constricted, thus giving rise to its name. Now, what this effectively means is that this is the place where the centrosome is present, or it is the center of the chromosome. Now, this necessarily does not need to be in the middle. Why so? Because depending on where it is present, you can see that the chromosomes have been labeled in different ways. So the first is metacentric, which is where the primary constriction is present exactly at the center. Second is submetacentric, which is slightly shifted from the center of the chromosome, and it is now somewhat inclined towards one of the two ends. So it is submetacentric. Third is acrocentric, which is more towards one pole, particularly inclined towards one pole, and that is acrocentric. And the fourth is telocentric, where you will not be able to distinctly observe any primary constriction along its end. And it would mean that the primary constriction is effectively present towards the end of the chromosome, towards the telomere. That effectively gives rise to the name telocentric. So these are the different arrangements that can actually occurred for different chromosomes, and the appearance of the same can vary depending on what is the organization of the genetic information of that is present on the chromosome. Now, why is this important? One reason for this is that during the beta phase, it does not necessarily mean that all the kinetic or microtubules will attach at the same position on the two sister chromatids. Depending upon where the primary constriction is present, kinetochores can attach towards one end, towards the center, a little away from the center, like we saw the different subtypes of these chromosomes. They will attach at different positions, and depending upon where they would attach, you would be able to see that there is pulling apart of the two sister chromatids in the two directions by the mitotic spindles assembled at the opposite poles of a dividing cell. So that is what we see in the schematic that was with reference to metaphase. Now, next comes the concept of condensins. Now, why are condensins important? Like we have just said, that condensin is what is keeping these two sister chromatids. So for them to be held together, the reason that these two sister chromatids are held together under normal circumstances and are only pulled apart during a certain stage of the cell cycle, particularly during anaphase, after metaphase assembly during the previous phase, they are only segregated after after a regulated mechanism is operating to make sure that the glue that is holding them together is being removed. So the glue in this case that is holding these two sister chromatids together is basically condensin. So condensin along with another complex of cohesin keeps this entire complex together. So if you look at the legend, condensin is a 5 subunit protein complex that is very similar to cohesin. It has two major subunits, the SMC2 and SMC4, which are also represented in the figures on the right. They somewhat look, they form this almost sort of circular structure. Sorry, it is similar to the way in which you would say the beta clamps that we talked about during the process of replication, that they were able to clamp onto DNA, or they were able to snap around DNA and hold it in place, so that the clamp loader was able to hold DNA in place, so that the clamps, beta clamps, could come in and they could do whatever job they had to do. So this particular structure is also being maintained by SMC2, SMC4 proteins. So SMC in this case stands for structural maintenance of chromosomes, which are a whole other group of proteins that are extensively studied to understand the process of cell replication, particularly cell division for mitosis and meiosis. And condensin is made up of these SMC group of proteins majorly, and it is also held together by three additional subunits, CAPG, CAPH, and CAPD2. Next, what you see at the bottom is that condensin is responsible for the restructuring and compaction of chromosomal DNA. But while it is not exactly clear how it is doing so, this is what you see as a representation at the bottom is the hypothesized way. Of course, this is part of a book. If you look at current papers, a lot more is understood about the way in which compaction and condensing is performed by these protein complexes. But according to the schematic, what we can see is that in the initial schematic representation, you are able to see that there is a little bit of looping of the chromosomal material, or the chromatin, which is represented in yellow, around which these little um, assemblies of the SMC group of proteins, or rather the condensin assembly, can be seen to snap around DNA while it is looping itself in an appropriate manner, which will allow for proper assembly of the condensin complex. Once it has done so, these individual condensin assemblies can interact with one another to form a sort of rosette-like structure. Now, what is rosette? Rosette basically means, if you think of some circles which are stacked one on top of the other, and that is forming a sort of continuous structure such that it looks uh, like a cylinder. So what it is effectively doing is each of these condensin assemblies of, I think, eight condensin assemblies are forming one floral structure that is followed by another and another and another on top. So what it does is the central core is made up of condensin assemblies, while the outer part has looping of DNA. So this entire complex is condensing the entire genetic information, also helped or assisted by other proteins, and that is uh, organizing the extensive genetic information into a much smaller space by compacting it extensively. So again, this is a very elaborate topic of discussion. I'm not going into details. This is not the only way in which compaction is being executed. There are multiple levels at which compaction is carried out. The most basal level is by nucleosomes, where you have the histone proteins around which DNA is getting wrapped. So an entire extended stretch of DNA gets reduced or occupies very little space by wrapping around nucleosomes. This is followed by subsequent steps where condensin comes in. It forms these individual rosettes. These rosettes are aligned one on top of the other, and so on and so forth. Effectively, that eventually leads to the formation of the chromosomes that we are finally able to see during prophase. So this again is an extensive domain of study. If you want, we can discuss this sometime later on. But for now, what we are majorly focused on is the step that, or is the protein that regulates this assembly in context of cell division or mitosis. So you can see the assembly or the manner in which this assembly is being carried out. So the reason that is important is because that will allow for segregation to be properly carried out by another particular protein known as separase. This is what regulates this entire process of cell division, that or particularly regulates the metaphase and anaphase steps of cell division, because cohesin holds these two sister chromatids together, and then a particular enzyme, which we'll come to later, separase, comes in and it separates these two. Now, this was with regard to the DNA or the genetic information. What also needs to be kept in mind is that there are multiple machineries which are also working in place, or that are working in at the same time to regulate the process of cell division in their own manner. How is that happening? That is because there is extensive role of the cytoskeleton, like we saw in those fluorescence micrographs as well, that the cytoskeleton has a huge role to play in context of cell division as well. In particular, the mitotic cytoskeletal machinery has the mitotic spindle, first of all, the mitotic spindle, and secondly, the contractile ring. The contractile ring, which is made up of actin and myosin, that eventually leads to the segregation from one cell into the two daughter cells. So the mitotic spindle has a huge role to play as well. So if you look at the schematic on the left, what you see are sister chromatids that are assembled at the very center or along the metaphase plate, like we just talked about, the equatorial plane or the metaphase plate of a dividing cell. And almost towards the center of each of these chromosomes, we can see blue colored kinetochore microtubules attached at the center of the chromosomes, particularly at the kinetochore of each of the two sister chromatids. And you can see that the orientation of them is back to back, so that microtubules adhere towards the opposite or towards the opposite ends, or they attach to the correct in the correct orientation with each of the sister chromatids. But what needs to be kept in mind is that these are not the only microtubules that are present. You would also be able to see a number of other ones. So first of all, we are able to see some in red that we also saw during the in the schematic for telophase as well, that there are some red colored microtubules, otherwise known as interpolar microtubules. These are microtubules that are present towards the or in between the two spindle poles, and they overlap with each other, and they are bound by certain proteins known as motor proteins. When we say motor proteins, they are extensively studied in the domain for cytoskeleton. I have not elaborated that in details here. If you want, I can share a link for it. It was a part of the discussion in some of my other lectures previously. But basically, if I just give an overview of it, the cytoskeleton consists of three types of components: the microtubules, which are made up of tubulin. The second are actin filaments. So microtubules are known as thick filaments. Secondly, come the actin filaments, which are known as the thick filaments, and they are made up of actin monomers. And third are the intermediate filaments that have majorly been associated with some um, strength based or structural integrity based functions within the cell. So the thick filaments or the microtubules are normally associated with more of the processes of cell division and these transport of information or transport of cargo from one end to the other. The actin filaments are majorly present towards the um, outskirts of the cell or just beneath the cell membrane. They are very closely attached with the cell membrane or the plasma membrane, and intermediate filaments are present at different locations based on their function as well. For example, lamins that we talked about are a type of intermediate filaments only. So the presence and the function of each of these cytoskeletal proteins depends upon what context it is serving its purpose. Now, apart from these three components, what regulates the uh, passage of information from one end to the other are certain motor proteins. So motor proteins, as the name indicates, they function as um, uh, they are protein molecules that are constantly in motion from one end to another. Majorly, there are two types or two families of motor proteins that are present inside a cell. The first is the kinesin and the second is dynein, and they can move in different directions depending upon what is the orientation of the microtubule. So the orientation of microtubule is something that I'll come to in a little while. But for now, what you need to understand is that the black dots that are present in the schematic are representative of these motor proteins. So they can move along these microtubules in either direction depending upon what purpose they are serving. So apart from the kinetochore microtubules, we are able to see these interpolar microtubules, which are closely associated with the motor proteins. And there are also some astral microtubules. So astral, as the name indicates, is representative of star-like. So they are forming asters or star-like structures from the two mitotic spindles. And they are extending from the mitotic spindles towards the outside of the cell, not towards the center where the chromosomes are present, but towards the periphery of the cell. So this is the arrangement of microtubules that you can see inside a cell at the time of during the metaphase at the time of cell division. Um, Next, towards the center, what you can see is that there are, it's a electron micrograph of centrioles. Now, what exactly are centrioles? For that, look at the figure on the extreme right. Um, these green colored structures that you can see, these sort of tube-like structures that you can see that are forming a cylinder towards the center. These are known as centrioles. So one particular centrosome consists of two centrioles that are present in a perpendicular manner. So the first green centriole is present at a certain angle, and with respect to that, at a right angle, you can see the second centriole. These two centrioles are surrounded by sort of an amorphous material that represents the pericentriolar material, PCM. Pericentriolar means just adjacent to or um, associated with or related to or in close association with centrioles. So they are right next to the centrioles, and it is made up of a number of different proteins as well. The pericentriolar material is again another area of extensive research as well, because it is not completely understood. So these two centrioles and the associated pericentriolar material all together forms the centrosome that you can see in this first schematic representation as well, shown in green. So these are not very distinct entities, but these are present. And these two extreme ends of the mitotic spindles are essentially consist of these two centrioles and the associated pericentriolar material. Now, under a microscope, you can commonly see these two pericentriolar materials, which is what is present in the electron micrograph as well. And on the lower schematic, you can see that there is a difference in the way in which these centrioles are present during the G1, S, G2, and M phase. So initially, during the G1 phase, you can see that these two centrioles are present with an associated pericentriolar material as it's expected normally. So, excuse me. However, towards the end of the G1 phase, you can see that somewhat there is some detachment of the two centrioles and an increase in the pericentriolar material. This is followed by the S phase, during which you can see that each of these centrioles has duplicated and formed or is associated with the pericentriolar material. And finally, during the S phase, each of the duplicated centrioles are also segregated into two, and then they can move across into separate directions, and they can form the two poles of a dividing cell. So the centrioles and the pericentriolar material are duplicating and also segregating during the M phase, along with the chromosomes that are carrying the genetic information. And the correct coordination between these two events is also extremely crucial, because each of these steps are being maintained by a large number of checkpoints. So each of these checkpoints must be met at the correct time in order for a cell to proceed through the process of cell division correctly. So that is what we see here. Again, if at any point I am not being clear, please interrupt me and ask whatever questions you have, and I'll try to explain them. So now we had just talked about the microtubules, right? Excuse me. So what you can see here are some electron micrographs. Now, what exactly are electron micrographs? It means that these are images that are taken for a cell using an electron microscope. Now, these are not dynamic in nature. These are sectioned images of a cell from one particular point of time, and then it has been fixed and it has been bombarded with electrons, and the representative diffraction that has been observed has been represented in the form of an image. The reason why this is performed is because it gives very intricate details and is representative of the correct information inside a cell. In today's time, you would see a lot more detailing using other forms of microscopy. There are some super resolution microscopy techniques which involve the use of antibodies and also lasers in particular. So that has come up in recent times. What was previously understood to be the more evolved technique was confocal microscopy. So confocal microscopy used to give sections. So for example, again, let me take this into consideration. If you have a structure like this, it would if you could chop this into multiple sections, and then you can look at each section individually, followed by which you would uh, sort of glue all those sections together to form a 3D image of the object that you were studying, then that is what exactly confocal microscopy used to do. It used to look at each section individually and get information in depth. Now there have been further advancements in these techniques, in fluorescence microscopy techniques, and there are super resolution microscopy, for example, STORM. These are rather advanced topics. You can look them up if you are curious about it. But yeah, these techniques are being used in current times to get extensive details about cells and get them in um, an easy to understand way by staining them with certain colors or certain antibodies which fluoresce in a certain color, because electron micrographs can often be difficult to understand, and also they are only giving information about one plane compared to lasers, which can go through individual planes of the 3D object that you are studying and give us a lot more information. So the images that you see, because these were studied several decades before, the technique that was most advanced at that point of time was electron micrography. So that's what you see here. So on the leftmost diagram, you're able to see individual microtubules, which appear like sort of cylindrical sort of structures, versus the second diagram that you see is sort of, it looks like a beaded bracelet sort of a structure. What this effectively represents is the top or a section of a microtubule. If you could section it, this is what it would look like. So If this is your structure, then this portion of it, if this is a microtubule, then this portion of it is represented by the second electron micrograph. What it effectively consists of are 13 protofilaments. Protofilaments are, again, this is represented on the right, in the blue colored schematic. What you can see are individual uh, columns of protein molecules, which are repeating units of alpha beta alpha beta alpha beta dinucleotides. These alpha beta, not dinucleotides, alpha beta tubulin dimers. So alpha tubulin and beta tubulin, which are two proteins that are present inside us, inside the cells extensively, because they are part of microtubules and they are what compose microtubules basically. So alpha and beta tubulin dimers are, they get assembled in repeating units to form an entire protofilament. 13 such protofilaments form a cylindrical structure, and if you look at the diagram carefully, you would be able to see that it is not forming a complete cylinder. It's somewhat, um, there is a bit of a gap that is present between the two. It's not that it's not closing, but it is not present in perfect orientation with respect to each other. There's a little bit of a alignment difference between the first and the last protofilament that is assembling in the formation of the microtubule. So this is what effectively forms the thick filament that I just talked about. So these microtubules are made up of these alpha and beta tubulin dimers, which you can see. And like we talked about microtubule assembly and disassembly, why is that important? That is because microtubules have a property of dynamic instability. Again, I'm not going into extensive details because this is a whole other topic of cytoskeletons, and it is, again, it is very extensively discussed in one of my live interaction sessions from an introduction to cell biology course, also covered by Neptel. Um, if you want, I'll provide the link for that within the slides as well. So just to get an overview of what exactly is happening there. Sorry. Okay, so what exactly is happening there is that these individual microtubules, they go through something, a process which is known as dynamic instability. This was also part of the video lectures by Professor Shikhala Loria. So dynamic instability means that the property of a microtubule is to never be stable. It means to never be static. That if a microtubule is formed, it should not remain in that arrested or static state throughout the process of throughout the cell cycle basically. So what it has to do is that it has to polymerize, which means it has to add more and more alpha beta tubulin dimers if it is required to elongate the length of the microtubule, and if necessary it has to get rid of some of these dimers to form a discontinuous structure. So there is this assembly and disassembly, otherwise also known as polymerization and depolymerization of microtubules, that takes place, and that very strongly coordinates the process of the cell cycle and cell division. So this assembly and disassembly of microtubules, because it is heavily dependent upon the polymerization and depolymerization process, therefore a number of drugs which are very late stage drugs used for cancer, for example, Taxol, otherwise known as paclitaxel, or platinum based drugs such as cisplatin, they actually arrest this ability of microtubules to undergo dynamic instability, and they make them static, which means that they would not be able to undergo this assembly disassembly process, and therefore the cell would be arrested in the step of division and will not be able to move forward from it. However, because of the mode of action, which is just to arrest any cell which is in the dividing state, which means that it is a non-specific way of arresting cells. That means if a particular individual, if a cancer patient is treated with drugs like these microtubule based drugs, then such a patient, upon ingesting or upon being injected with drugs like these, some drugs are oral in nature, some drugs can be IV injected drugs, so depending upon whatever route of administration is most feasible for that drug, what it will effectively do is that that particular drug can go and bind to or can act upon any cell of the body, any dividing cell of the body. If that happens, then such a patient will have rapid inhibition of cell division in any cell, or any dividing cell within their system. For example, one of the major cells that are affected are within the hair follicles, which is why patients that undergo chemotherapy, particularly by these drugs, they often get affected and they lose their hair because their hair cells or the hair follicles are very largely affected, and therefore their hair follicles are no longer able to divide as long as the chemotherapeutic treatment is continuing. Um, they also become weak. Their immunity is severely compromised because their immune system can also get very largely affected by this particular process. However, it is the only reason that drugs like these are at all entertained or at all considered for treatment is because the patient has reached a state where the doctor usually looks at whatever is the greater good, that even if the patient is losing hair or is getting affected in some other sort of collateral damages occurring in such a patient, it is still acceptable or it can still be afforded because the patient is surviving in the long run. While these statistics are not very favorable, it does so happen that patients after being treated with these drugs have managed to survive, and that is why, or at least extend their lifespan by a number of years, by a large number of years. So patients go for such treatment in this direction. So what I'm trying to bring your attention to is that just studying one particular protein and the way in which it works inside a cell and the function that it performs inside a cell can give you a target or a way in which you can treat diseases as severe as cancer. So yeah, that was about microtubules. Now, what you see at the bottom is that for a chromosome that has completed replication, the kinetochore microtubules are attaching towards the centromere, or what we talked about as the primary constriction, which is present along the length of a chromosome. Sorry. And from there, the chromosomes, the individual sister chromatids from the duplicated chromosome will be pulled apart in opposite directions during the anaphase. And again, the electron micrograph on the right is representing a similar thing. What you can see is an anaphase chromatid. The huge like I don't know how to explain it, but basically this extensive crowding that you can see, or these gray sort of speckle-like structures that you can see, that is basically the genetic material being wrapped around by all of these condensing proteins or condenses and histones, etc. So that is the extensive um, protein and a nucleoprotein complex, which is DNA and protein complex that is forming the entire chromosome. So this V structure that is present is representative of an anaphase chromosome, and what is present towards the right is the kinetochore portion to which you can see individual microtubules are embedded or attached, and this determines that the direction in which this chromatid, the sister chromatid, will move is towards that towards the right, which is towards the direction in which the microtubules have been embedded. So this is what we talked about, that this step is essential for cells to move in the opposite direction. Now, if you remember, we had said that the protein that holds these sister chromatids together, one of the machineries that holds the protein, um, or rather holds the two sister chromatids together, is condensins, and what separates them is separase. So during the anaphase transition or metaphase to anaphase transition, that after all of the chromosomes have aligned and now they need to segregate between these two stages or during the transition from one stage to the next, what comes into play is separase. So if you pay attention to the diagram here, you can see that during the G2 phase, which is just before the M phase, you can see that the two sister chromatids are being held together by the cohesin complexes, which are keeping them very close to each other. However, what you can see is that during the metaphase stage, there is action of MCDK, which is just or which is increasing in number or increasing in expression because the cell is about to enter into mitosis or is about to enter into the step of cell division. The increase in MCDK is activating the APCC complex. Now, the APCC and the SCF complexes is something we have already discussed, I think, in week 2, because these are part of the proteasome machineries that are present normally in cells but have huge roles to play during the process of cell division. Particularly, APCC is very relevant during these metaphase to anaphase transition. So, the activation of the APCC complex is carried out by CDC20 and by MCDK, uh, upon which CDC20 binds. And what that controls is that it releases securin and separase complexes. So under normal circumstances, if you just remember the names, it would clear things for you. What protein machinery or what proteins hold chromosomes together or condense chromosomes or condense chromatids are known as condensins. However, what separates them or sort of cleaves the interaction or cleaves the attachment between the two is separase. It is an enzyme because it is cleaving these proteins and separating them in opposite directions for allowing the movement of chromosomes or chromatids in opposite directions. So separase is the enzyme that will act in this direction. However, if separase is always present in an accessible manner for the cell to within the cell for it to go and execute its activity, then that is very detrimental and chromosomal assembly will never be able to successfully happen. Therefore, inside the cell, under normal circumstances, prior to this metaphase to anaphase transition, the separase enzyme is held or is sequestered. The meaning of sequestered is that it is held or it is inhibited from executing its activity by another protein, which is known as securin. Securin secures separase from acting on condensins. So you can just make a little flowchart in your mind for clarity. So at the top is securin, which is binding to separase and keeping it sequestered. When securin is no longer bound, it is removed, then separase is ready to execute its activity. Separase is coming and digesting condensin, and that is separating the two chromosomes. So that is what you can see here. There is ubiquitination and degradation of securin by active APCC complex. And what that effectively does is that it activates separase. Then separase can act upon the metaphase chromosomes and it can cleave or dissociate cohesins, which will allow the two sister chromatids to move in opposite directions. And that will complete the transition from metaphase towards anaphase. Just a second. Now, what you see on the right, I think this also is something that I mentioned in one of the previous classes. Um, that one of the major checkpoints inside the cell to avoid non-disjunction is something that occurs post or somewhere between this transition from metaphase to anaphase after alignment has occurred, post alignment, before separation towards the opposite poles. The cell has to make sure that each and every chromatid or every sister chromatid from each and every chromosome is attached to a kinetochore microtubule, and in fact it is attached to opposite kinetochore microtubules from two different mitotic spindles of the two poles of the dividing cell. So what you can see here is a particular protein that actually ensures this. So there is a signal basically that is in place, which is known as MAD2. MAD2 kinase is a signal that makes sure or that declares to the cell that yes, each and every chromosome or each and every chromatid has been attached to a kinetochore microtubule. Let me just read out the legend for this. So the initiation of sister chromatid separation is carried out by APCC, which is what we said for the figure on the left. It leads to the ubiquitination and degradation of securin, which normally holds separase in an active state. The destruction of securin allows for separase to cleave SEC1, which is part of the cohesin subunit complex that holds the two sister chromatids together. Then the mitotic spindle has pulling forces that pulls these two sister chromatids apart. Additionally, in animal cells, there is phosphorylation by some CDKs also that can normally inhibit separase, which has not been shown in the diagram. Therefore, CDK inactivation in anaphase also promotes the separase activation by allowing its dephosphorylation. That is representing what exactly happens at the end of the chromosomal separation. Now, simultaneously, what happens is that there are MAT2 proteins that are MAT2 phosphorylation that is also controlling the process of metaphase to anaphase transition. What you can see on the fluorescence micrograph on the right is these sort of red colored two, if I don't know if it is clear, but there are two particular little dots that you can see towards the bottom that is representative of one particular chromosome that has yet not attached to any microtubule. You can see that it is not exactly aligned along the metaphase plate, and therefore no microtubule has been able to adhere to it. Whereas for all of the other chromosomes which are represented in blue, you can see that they have attached to individual microtubules and therefore they are not giving out any negative signals. So if a cell is sensing that there is still one particular chromosome which has not attached to a microtubule, it means that this particular chromosome could go in either direction, and therefore only one of the two daughter cells would receive a copy of this chromosome, while while the other daughter cell will not receive the same. So that would be a non-disjunction of chromosomes, or unequal segregation of the genetic information among the two daughter cells, and that could be disastrous for the cell in the long run. Therefore, the presence of MAT2 kinase sorry, ensures that as long as a cell is in a condition that it is still having an unattached chromosome, or a chromosome that is unattached or yet not attached to a kinetochore microtubule, such a cell will not be able to or will not be allowed to transition into anaphase and would be arrested at the metaphase stage. So therefore the cell has kept this checkpoint mechanism in place in order to make sure that only equal division, under normal circumstances, only equal division of chromosomes occurs. Despite all of these checkpoint mechanisms, of course, there are errors that occur, and cells often are able to skip these, or cells are not often able to overcome these errors or these fallacies. And despite all of these problems, they still are able to duplicate, and that is what leads to all of the different types of diseases that we talked about. Yeah. So on the left, what you can see is the representation of anaphase A and anaphase B, which is the result of all of these coordinated processes occurring in concordance with each other. So you can see that during anaphase A, there is the movement of these microtubules. First, there is alignment during the metaphase, and after alignment has occurred and correct attachment of the kinetochore microtubule has also occurred, you can see that they will start migrating in either direction. The chromosomes will start migrating, kinetochore microtubules will become smaller and smaller. The the condensin complex would be cleaved by the separate securase regulation mechanism that we just talked about, and there would be migration or movement of the sister chromatids towards opposite ends. Also, what you see is that during anaphase B, the two spindle assemblies, or the two mitotic spindles, would also move apart in opposite directions, and that would further lead to elongation of the cell for it to allowing the cell to enter into telophase, and that would be followed up by cytokinesis. So what you see in the figure present at the bottom is that for a cell which is undergoing such a process, or the process of cytokinesis in particular, I think there's some error. Someone has a question. Deepthi, yes, go ahead.
Yes, ma'am. Good evening.
Yeah, fine.
Ma'am, you in the previous slide you have mentioned that if the cell is arrested at the before to go for the metaphase, if it is not, the chromosomes are not attached to the spindle fiber. So
Right.
when it is arrested, whether there is a mechanism to to repair it, or for the attachment of the chromosome, because that particular genetic information will not go further. So how it
Right.
is regulated?
So, correct. So, a cell will always try to, in general, the principle of any cell is to always try and conserve energy. Now, as you understand, this entire process of duplication and whatever steps have been taken till now have involved a huge expenditure of ATP for the cell, right? Synthesis of so many proteins, all this regulatory mechanisms, etc. So a cell will always try to salvage whatever energy it has expended, and it will try to repair it. It will try to it will wait till each of the chromosomes have attached. So there are different ways in which this can occur. Either it will disassemble all of the microtubules and it will go into a rearrangement of the chromatid, but again that would be a huge expenditure of energy. What it will effectively try to do is that it will try to bring about a separate microtubule, because microtubule assembly is not that expensive. But I would have to look into this in details. But if you look at it from a energy conservation perspective, the cell will try to save whatever energy it has expended, and it will try to wait until each of the chromosomes has attached with a new microtubule or with a kinetochore microtubule, and then it will push that cell towards replication, towards cell division. However, what I said about metaphase arrest is being maintained by microtubule targeting drugs. So, in such cases, because the microtubule is frozen in a state that it cannot assemble or disassemble, under such circumstances, such a cell enters a state of arrest. It can neither divide nor it can proliferate. It cannot do anything, and it is stuck in that state. And eventually, such a cell will be taken by the either would be removed by apoptosis or would be identified as a defective cell by the immune system and would eventually be eliminated. So, a metaphase arrest is a more serious problem that is encountered when microtubule targeting drugs are taken, versus when a cell is in this stage where it is keeping this as a checkpoint mechanism. It will try to repair the error and will try to take that cell forward for division to be completed. So I hope it was clear, Dipti.
Yes, ma'am. It is clear. Thank you so much.
No problem. Okay, so yeah, so that is what we see here for anaphase and the processes of or the substeps of anaphase A and anaphase B. Following this, we see that if you remember initially, we had seen that there are two cytoskeletal machineries that operate during the cell cycle. One is the mitotic spindle, which we have extensively discussed, and second is the contractile ring. I have not given much details about it. Most of it was discussed well in the lectures, but basically, what effectively happens is a part of the not part of the cytoskeletal topic only, because actin and myosin. Actin is, of course, the thin filament, a part of the cytoskeleton, and another protein which is associated with it is myosin. And this actin myosin assembly plays a huge role in contraction and relaxation within a living system at large. For example, each of your muscular movements that any of us are able to execute is uh, because of the sliding there is a theory known as a sliding filament theory, which is the movement of myosin. So if you look at these as actin filaments, there are actin filaments which are arranged one after the other, and there are myosins which move along it. So this relative motion of myosin and actin with respect to each other is what generates the force that is required for a muscle to contract or relax. Each and every muscle in your body, your eye muscle, your arms, and whatever you can think of, for even your esophageal sphincter to work, or for food to move down your entire digestive tract. So all of these are regulated by contraction and relaxation, which is mediated by actin and myosin. So they have a global role to play in an entire living system, which is very extensive. In context of cell division, this particular or this complex or these two proteins are responsible for the force that is required to segregate the cytoplasm of a dividing cell from one into two. So there is an entire bundle, or why we are calling it a bundle is because the actin filaments are able to assemble in two different ways or organize themselves in two different ways. One way is in the form of actin bundles where they are oriented one after the other. So actin filaments are arranged one after the other, versus the other organization of actin is in the form of a mesh like network where they can sort of form a, like you understand, a net like structure or a mesh like structure. So actin bundles and actin meshes can form in different manners. In context of cytokinesis, actin is forming a bundle along with myosin, and it is forming a huge um, sort of an elastic, it is forming like a strong rubber like structure which will pinch off these two daughter cells, or will pinch off the cytoplasm, or will separate the cytoplasm between these two daughter cells in order to separate the centrioles and the plasma membrane and all of these organelles that are present. What this is actually something that we keep on forgetting, that because cell division so largely focuses on only the nuclear division and the chromosome duplication, chromosome segregation, which is of course important, what needs to be kept in mind is that the cell is not just made up of the nucleus. The cytoplasm has everything outside of the nucleus, right? So each of the organelles, the mitochondria, the Golgi vesicles, the lysosomes, all of that also undergoes duplication. Most of all, the plasma membrane, which is at the outside of a cell. So the boundary of a cell, of an individual cell, whatever extent of boundary one particular cell has, is not enough for division into two cells. So that boundary also has to divide. So there has to be lipid synthesis, and there has to be extension of that boundary as well, following which it will divide into two. So all of these mechanisms are also kept in check, and once they have been checked, only then the contractile ring that is present will come into play. It will first, there is some invagination which occurs, which brings about or which establishes the point from which cells will divide from one into two, and following that the pinching off mechanism is what separates them into two independent daughter cells. So that is what you see on the left. Apart from this, what you see on the right are so that was basically the process of cytokinesis, which was once again initially studied in yeast. We have established this extensively, that yeast are the most preferred model of studying cell division, particularly in eukaryotes, because of the conserved nature of the proteins that are present. So while they are not exactly the same, there are analogs or human analogs and yeast analogs that are present, and therefore they are a good model to get a basic idea of the step that we are interested in. So in the initial studies that were carried out in yeast, it was seen that cytokinesis has a preparatory period during which there is an initial invagination that is occurring and the formation of a contractile ring, and finally an execution period during which you can see that there is further invagination and complete separation from one cell into two cells. Now, what needs to be kept in mind is that yeast does not have equal segregation of the cytoplasmic material. It forms buds. So the mother cell is much larger, and from there the bud that originates is much smaller, and that grows in size, and then it can undergo cell division. However, for mammalian cells, what you see is that there is that the daughter cell is the equal size of the initial parental cell. So there is equal separation or equal division of the cytoplasmic material between the two cells in this case. And apart from the nuclear material, you can see that each of the cells have their own centriole copies as well. They will also undergo duplication. And at the center, you can see the alignment of the microtubules, the actin and myosin complexes at the center, which is forming the contractile bundle that keeps on decreasing in, or that keeps on increasing the interaction, thereby decreasing the zone of association, and eventually it comes to a complete standstill and it separates into two independent daughter cells. So that is the process of cytokinesis. So with this, what we are basically covering is that I have not included the final lecture because it was rather brief, and you can go through it. It basically looked at the way in which centrioles and the concept of centrosomes have evolved across different organisms. But basically, because the idea for today was to understand the process of mitosis, the sub steps of mitosis, followed by these the concept of cytokinesis and what exactly operates in that regard. Therefore, that is what we have covered in the basic lectures. So again, for reference, please go through Bruce Albert's. It is one of the best books you will find for any details about cell division, cell cycle, and basically any molecular biology topic. And if you want specific details on the cytoskeleton, there are two books. There is Cell Biology by Gerald Karp, and there is another book by Cooper, George M. Cooper, I think. So yeah, these are two excellent cell cycle, cell biology based books. They do not have much detail about cell cycle because it's more of a molecular biology topic, whereas cytoskeleton is majorly a cell biology topic. So you will find these extensively mentioned in these books. You'll be able to get a lot more detail about each of the cytoskeletal components if that is something that interests you. And again, if you want, I can give some material in that regard as well. So with this, we are completing the topics of discussion. I will now show some sample questions based on the previous year's um, question that was mentioned. If there is any doubt, please go ahead and ask. I'll try to answer that. Just a second, let me reshare my screen. Okay, so yeah, I hope it is visible. Yeah, I think it's visible now. Okay, so just for some sample questions, based on whatever topics we have just discussed. Um, so the first question is that which of the following kinases is not required for mitotic events to take place? So while this is not something that I directly mentioned in the interaction session, This was actually a part of the video lectures if you have listened to them carefully. So apart from the general machinery that operates, you would see that there are a number of other proteins also that function and that mediate the appropriate regulation of the entire mitosis, or the submitotic steps and the entire process of mitosis, and that includes polokinases, that includes ORCAs or aurora. So Aurora A, Aurora B are kinases that are represented as AURKA or AURKB. So they are known as ORCA and ORCA-B. So these two kinases are extensively required along with polo-like kinases as well for mitosis to be triggered and for mitosis to be properly regulated. So since they are required, the correct answer for the question is none of the above, because these are all necessary for mitotic events to take place. Second is that the key event that occurs at the metaphase to anaphase transition is which of the following? So first of all, sister chromatid dissociation and segregation, sister chromatid association on the equatorial plane, sister chromatid condensation, or all of the above. So sister chromatid condensation, as we have extensively understood, it is part of the prophase step, because that is where the assembly or the organization of the entire genetic material takes place. So that is not a part of the metaphase to anaphase transition, certainly not. The second option is that sister chromatid association along the equatorial plane. That is definitive or is the characteristic feature of the metaphase step where we see that they all of the chromosomes they align along the center of the cell, or the equatorial plane of the cell. And the first option is representative of anaphase, metaphase to anaphase transition, which is that there is dissociation or separation and segregation of the two sister chromatids towards the opposite poles. And therefore, it is the key event that occurs during this transition from metaphase to anaphase. So option A is correct in that regard. Um, then comes sister chromatid cohesion is established in which step? G1, S, metaphase, anaphase. So if there is any lack of clarity, because this is not something that we have directly discussed, if there's a lack of clarity, always go by elimination of incorrect options. So if we look at the options one by one, if you see anaphase, of course, that is not where sister chromatid cohesion is occurring, because that is where segregation or separation or dissociation takes place. So that is certainly sorry, that is certainly not the correct option. Then comes metaphase. So we have seen that pre-assembled or chromosomes that already have two aligned sister chromatids, they just orient themselves around the metaphase plane. So that is also not where cohesion is taking place. G1 phase is the pre-replicative phase. So there is, in that stage, there is only one copy of each of the sister chromatids. So of course, there cannot be any cohesion during that stage. So that automatically brings us option two, which is that during the S phase, when replication is occurring and one copy of each of the chromosomes is being duplicated into two, during that stage only we are able to see that there is cohesion or there is um, association of the two sister chromatids with one another, because that cell has committed to replication and will further be proceeded or will further go into the process of division. So that makes option two correct, or the S phase is the correct option for such this question. Which of the following class of proteins are not present in the pericentriolar material? Okay, I did not discuss the PCM composition, but basically they have cell cycle regulators, they have signaling molecules, and they certainly have microtubule organizers, because from there the microtubules can originate and the mitotic spindle assembly can take place. So all of these three are prerequisites of the or compose the pericentriolar material. So those are the correct. All three of them are correct. Therefore, none of the above is dispensable for pericentriolar material, and that makes the fourth option correct. Um, Microtubules that overlap with each other when in anti-parallel orientation are called as, if you remember that diagram that we saw of the different types of microtubules, then those that overlap with each other and are associated with motor proteins are kinetochore microtubules, which is they're interpolar microtubules. I think the options were repeated in this case. So yeah, Okay, no, they're not repeated. Sorry. So there's interpolar and there is intrapolar. There is nothing called as intrapolar microtubule because within a pole you would not necessarily have microtubules. There are astral microtubules which originate from one pole towards the cell periphery, or there are kinetochore microtubules which associate with the chromosomes. Sorry. But those that overlap with each other are interpolar microtubules. So that is the correct option. Which of the following proteins is absolutely required for the metaphase to anaphase transition? Helicase, separase, glutaminase, and phosphatase. So, of these, of course, separase is the correct option because that is cleaving the cohesin complexes, that is allowing for the two sister chromatids to detach from each other. So, that is the correct option. The spindle pole body that is present in budding yeast is equivalent to which of the higher animal or mammalian structures? Centrosomes, lysosomes, Golgi body, and ER membrane. So, of course, the spindle pole is equivalent to centrosomes in case of mammalian cells because that is the region from where you can see that there is the attachment with the mitotic spindle. So, centrosomes and spindle poles are analogs or equivalents of each other. So, that would be the correct option. Then the FRAP experiment. So this was also a part of the lectures. The fluorescence recovery after photo bleaching was an experiment that was performed to understand that microtubules have dynamic instability under normal circumstances within a dividing cell. So the FRAP experiments on microtubules yields which of the following inferences? The options were A, individual microtubules are dynamic and their subunits are being constantly replaced. They are constantly polymerizing at both ends. Tubulin remains at the equatorial plane of the cell cycle, or microtubules are static and stable in nature. So option 4 is of course not correct. Tubulin remains at the equatorial plate of the cell cycle is also not correct. Only chromosomes are at the center. Individual microtubules are constantly polymerizing at both ends is also not correct. Polymerization is very orientation specific. So microtubules have a plus end and a minus end, and they are only adding only adding new dimers in the dividing end only. So the FRAP experiment was representative of option A, that if you basically the experiment intended to shine laser or in put laser or direct laser in one particular region of a cell so that in those regions whenever laser is incident upon fluorophores in one particular region for a certain duration, it causes photo bleaching, which means that these fluorophores will not be able to fluoresce again. They have been bleached. So after photo bleaching, if there is recovery of fluorescence, it means that there has been some rearrangement that has occurred, which has allowed the tubulin units from other regions or some rearrangement from other tubulins, which has allowed the restoration of fluorescence for microtubules, and therefore it indicated that microtubules are dynamic in nature and their subunits are constantly in circulation. They are being replaced. So that was the correct option. And the next question was that individual microtubules, sorry, the next question is for selecting the correct statement regarding spindle attachment to chromosomes. So first option is that sister kinetochores are assembled in a back-to-back orientation to reduce the chance of mono-orientation. That is certainly correct. Bi-orientation is required and is necessary for correct segregation between the two spindle poles. Second, cohesion at the centromere makes the paired sister chromatids to be more fluid in nature. No. So cohesion actually keeps these two intact and be less fluid and in fact be more rigid, which induces more tension at the centromere, which allows the attached microtubules to pull them apart in opposite direction. So there is some biomechanics which is also at play in order to execute this process of sister chromatid movement or microtubule flux that executes this separation between the two sister chromatids. And third option is that bi-orientation of chromosomes is important to generate tension at the kinetochore between the sister chromatids. That is, of course, true. Therefore, option 1 and option 3 are correct. Or statement 1 and statement 3 are correct, which makes the third option correct in this regard. And finally, for the last question, which is very specific and it's not something that I mentioned. It is part of the mitosis exit network, which is MEN. So there is a complex of proteins that are operating during this step. So the extended binding of NET1 to CDC14 can lead to which of the following? So this NET1 CDC14 complex has a role to play in mediating the exit of a cell from mitosis. So if there is extended binding of these two, then it can actually delay the exit of cells from mitosis. So that would make the option 2 correct, which is that delay in mitosis exit is what would happen in that case. So with this, I come to an end for the questions, sample questions for this week as well. So to sum it up, what we basically looked at are the steps of, so first of all, we looked at the previous week's assignment, went through the questions, we went through whatever topics were discussed, including mitosis and in extensive detail, the sub steps, the regulatory steps, and also, of course, the process of cytokinesis, what mediates it, and some extra information regarding the same as well. So I'll follow with some sample questions for the next week, which I hope would be helpful in figuring out how to answer your assignment questions as well. And with this, I think I can conclude the session from my end. If there are any questions, please bring this up and then we can end the class for today. If there are no questions, then thank you for joining the session for today. Uh, if you have any other questions, you can post them on the discussion forum, or you can attend the other interaction sessions as well, or you can join the next interaction session, which will be on Sunday, which is the normal schedule that I follow. So you can join that session as well from 4 to 6, and we will be discussing the next week's topics, which is for week 5, and we'll also go over some sample questions for the same as well. So thank you so much for joining. And if you have any doubts, please bring them forward. And if you have any suggestions, please mention those as well. And yeah, I hope you have a good night. Thank you so much for joining. Bye bye.